So I kind of got cut off, but what I was saying was when the DNA condenses into the chromosome, it's easier to move it around during cell division. All right, so before a cell divides, the DNA, or you could say chromosome, needs to duplicate. So the DNA is copied. And when the DNA is copied, we call these two copies sister chromatids. Now, these sister chromatids are exactly the same. They have the exact same information. So this is what happens when DNA is copied. And there's a whole big process called DNA replication that we'll learn about. But basically, you take a DNA molecule, you pull it in half, and then you fill in whatever you're missing. And that's how you make a new molecule of DNA. So the new molecules of DNA are actually half old and half new. But either way, these two molecules together are called sister chromatids, and they're identical. They're joined together in a region in the middle of the structure, which is called the centromere. So the centromere kind of just holds together the sister chromatids. Now, during cell division, we're going to take those sister chromatids and split them apart because think about the whole reason why we copied our DNA. We copied our DNA because we're making new cells. And if you're going to make a new cell, well, guess what? That new cell also needs DNA. So you have one molecule of DNA that you start with. You copy it. Now you have two molecules of DNA. And now you pull them apart. Because even though they're together when they're first duplicated, that doesn't last very long. What we want to do is pull them apart and send them to the two separate daughter cells. Okay, so this picture might help explain a little bit better. So we'll start with one chromosome. We'll go through the DNA replication process. This happens during S phase of the cell cycle. And what we end up with is one big chromosome that's actually made up of two identical molecules, which are called sister chromatids. These are identical molecules of DNA, and they're connected. But when we split apart the cell into two new cells, we're going to pull these sister chromatids apart. That's what happens during anaphase. So what is the cell cycle? The cell cycle is just like the life cycle of the cell. So it starts here. This is the new cell. And this new cell is going to go around this cycle clockwise, and it's going to go through some different phases. So after we split some cells, we get a new cell. But new doesn't really make sense because it's not really new, it's part old cell. The old cell splits apart into two new cells. So we have this more accurate term, makes more sense. It's called daughter cell. So let's say this is a daughter cell, it just split apart. The first stage it will go through is G1 or gap 1. And during the G1 stage, the cell is going to grow and it's going to do its normal job. Remember, cells, their job is not just to divide. That's actually a small part of their life, maybe like 10% of their life. Their main job is to do cell stuff that we've been learning about all year. So if it's a skin cell, its main job is to do skin stuff. And if it's an eye cell, its main job is to help you see. And if it's a stomach cell, its main job is to do stomach things like helping you digest food. Division is a small part of the cell's life. Okay, so during G1, the cell is doing its normal job. Then it goes into S phase, which is about half of its life is S phase. And during S phase, there's a couple things happening. First of all, it's still doing its normal job. This is a normal part of the cell's life, okay? It's still living its normal life. But it's also doing S, synthesis, DNA synthesis. During S phase, the cell is making more DNA. That's what we were just talking about before.